Hello everyone, myself Sia Vishwesh Naik from TY BSc B.Ed welcome you all to my presentation on epidermal hard derivatives, wherein we'll focus on digital cornification in amniotes, that is the claws. Now, before moving further, let's take a close look at the integumentary system. The skin of all vertebrates are built accordingly with the same basic plan. The set of organ system forming the outermost layer of an animal's body is the integumentary system. It comprises the skin and its appendages, which act as a physical barrier between external and internal environment that serves to protect and maintain the body temperature of the animal. Integument is a complex structure comprising of three main layers: epidermis, dermis, and hypodermis. The first layer, that is the epidermis, is further divided into several layers. They are stratum corneum, stratum lucidum, stratum granulosum, stratum spinosum, lastly stratum germinativum. The outgrowths are mostly by the stratum corneum layer. Now let's study the derivatives of integuments in detail. Depending upon the layer of origin, it is divided into two types. First, epidermal derivatives. They are the derivatives formed from the epidermal layer. Hence the name is epidermal derivatives. Whereas the dermal derivatives means that the derivatives are derived from the dermis layer, which is the second layer. They are further categorized into hard and soft derivatives. Here, dermal derivatives are only of hard type of derivatives, whereas epidermal derivatives can be of both the types, that is, hard as well as soft type of derivatives. In this video, we will learn the epidermal hard derivatives, that is the digital cornification in animals. These digital cornification are the formation of the horny layers over the digits. These horny layers are of stratum corneum layer. As you can see in the flow chart, it can be in the form of claws, nails, hooves, hair, but only feathers are originated from the stratum germinativum layer. So, how does this process of epidermal hard derivative take place? The epidermal hard derivatives or the digital hyperkeratosis grows parallel to the surface of the skin. These are the outgrowths formed on the digits of an animal. These type of derivatives comprises of two plates. They are the dorsal plate or the unguis and the ventral plate or the subunguis. In this picture, you can see a basic structure of the nail bed where the upper part is the unguis and the lower part is the subunguis. The dorsal plates are the compact horns or the nail plates, whereas the ventral plates are the sole horns or the sole plates. Now let's take a close look at claws. As you can see in these pictures, claws are narrow curved structures curled downwards from the edges of the digits. These are present in amniote classes. Here, amniote are referred to the group of tetrapod vertebrates that includes all reptiles, birds, mammals and their extinct relatives. The word amniote is derived from the word amnion, which refers to the development of the protective membrane around the embryo. These are the modification of stratum corneum layer. Claws are the epidermal derivatives hardened by the keratin protein, and these claws had first appeared in class Reptilia. Claws are seen in reptiles, few amphibians, birds as well as in mammals. Now let's see the claws in case of amphibians. This process is very rarely seen in case of amphibians. The only exception is in the case of South African toad, that is Xenopus laevius, wherein we find some cornified endings in the digits of these toes. You can see in the diagram as well as in the picture. Please take a note here that claws are not typically seen in case of amphibians. Xenopus laevius is the only exception. These South African toad have webbed feet and horny claws on 3 toes. Now that we have seen amphibian claws, let's understand the reptilian claws in detail. Let's see how are these claws different from the birds and mammals. As we can see here in the structure of reptilian claws, we have the dorsal plate, the unguis, and the ventral plate, the subunguis. Here, unguis is sharply curved and below this unguis is the subunguis. This claw is supported internally by the terminal phalanx. Here it is given as the tapering phalanx, but both are same. What exactly is this tapering phalanx or phalanx? It is a bone supporting the claw and can be present in the finger or the toe. Moving further, now you can see here a fold of skin or the invagination of skin that leads to the claw root. This is the claw root. Externally, the claw looks like this, but internally it looks somewhat like this, where the phalanx provides the structural support, flexibility, and a foundation for keratinization. Now that we have seen the reptilian claws, now let's take a look at avian claws, that is the claws of the birds. It is also called as talons. Talons are sharp, hooked claws only seen in the birds of prey. They are long and sharp as compared to the ordinary claws of the birds. They are further of many types. In case of sparrow, the claws are designed for perching. In case of owls and other prey birds, they are for striking and grasping the prey. Birds show typical reptilian claws where we see that the unguis is long and it covers the subunguis. Moving further, let's see the claws in case of mammals or mammalian claws. Now here the example is of rabbit. The claws are shorter than that of the reptilian and avian claws. Here we can see that the unguis is covering the subunguis and this subunguis is very much reduced and is continuous with a fleshy pad. This pad is also known as torus, which is present under the digit. In the next slide, we will see the claws in case of carnivorous mammals. The carnivore mammals possess the claws that are retractile, also known as the retractile claws. These claws can be hidden easily in the animal paws and can be withdrawn out whenever needed. So this process is taking place with the help of these digital tendons and the elastic ligaments. Since these claws are withdrawn inside, these claws remain very much sharp. These pictures are of the claws in case of mammals, that is the carnivorous mammals, wherein the first picture is of the retractile claws and the second picture are of non-retractile claws. As you can see here, the claws are blunt as they are retracted outside. They lose sharpness, hence they are a little blunt as compared to the retractile claws. These are the references. Thank you.